Okay, so I'm Andrew Buxton from the Durrell Institute for Conservation and Ecology at the University of Kent and Amphibian and Reptile Conservation. I'm going to talk about environmental DNA and how we are using it in various projects. You see I've got Freshwater Habitat Trust and SMNCP badges up there. So my work for Amphibian and Reptile Conservation, I'm seconded into a joint project with Freshwater Habitats Trust, which is known as the South Midlands Newt Conservation Partnership, and I'll get into what that is a bit later. Naomi, who works predominantly for Freshwater Habitats Trust, sends her apologies, but she isn't very well and she didn't want to infect everybody, so I got her slides at about 7 o'clock last night, so wish me luck with those ones. So I'm going to talk about initially what environmental DNA is and how we use it for surveys. I'm then going to go into, hopefully I'll have time to go into 3 of the different projects that we're working on as a group of organisations, and the data that we've got coming out. So how we're using eDNA with District Licencing, I'll go into what that is as well. There'll be PondNet programme, which among other things has a national surveillance element to it for great crested newts using volunteers and environmental DNA, and we are just starting to pick up the eDNA meta-barcoding route, which looks at biodiversity in freshwater habitats in a much broader sense than just single species. So what is environmental DNA? Well, aquatic environmental DNA is DNA that has become separated from a source organism and separated and suspended in the water column. Now, eDNA doesn't have to be aquatic. So as I'm standing here, I'm shedding skin cells which are at the moment falling to the floor, getting incorporated into this lovely carpet. Some are being taken away on the air, and you can isolate it from those environments as well. But aquatic samples, we survey for aquatic eDNA, collecting a bulk sample of water from your pond, your river, your stream, and then we use forensic level lab-based techniques to extract the DNA from those samples, bulk samples of material, to identify which species are using that environment. So, yeah, as I mentioned, we're going to concentrate on ponds and streams today. You collect a bulk sample of your pond water, going around collecting from each microhabitat that is within reach from the edge of the pond, being careful not to contaminate the samples with yourself, which generally means you can't get samples from the middle of the pond unless it's quite narrow. You combine all of your subsamples from around the pond into one plastic bag, which is sterile when you get it. And then that water that you've collected you need to preserve somehow. So there's 2 general ways of preserving your environmental DNA sample, either on filter paper, which you can push through a filter unit using a syringe, or you can pull it through the filter paper using a vacuum pump. These are then preserved with some chemicals which prevent the DNA from breaking down. The other way of preserving your sample is you take the water itself and you preserve that in very highly concentrated ethanol, and send the whole sample off to the lab. Once the laboratory gets it, the DNA in each sample is actually very, very weak. So you might have a handful of copies of each of the sequences that you're interested in a sample. So the lab needs to concentrate it down and get it off the filter paper if that's the method that has been used to collect it. And then they need to clean out all of the other gunk that has been collected with the sample. So you can imagine all of the suspended solids that's in the pond water. You've got all of your little clay particles in the water, etc. And you end up, after they've cleaned and extracted the DNA, with a small tube with a couple of 100 microlitres of liquid, and hopefully just the DNA that was collected earlier on in the mass bulk sample, without any of the inhibitors that stop the processes further down the stream working. The lab then have two routes they could take. They could either target a specific species that you're interested in, which is what we have been doing quite a lot with great crested newts. I'm sure a lot of you are aware of this, where if great crested newt DNA is in the sample, you put it into this PCR machine and you get a spike on the computer if great crested newt DNA is present. And there's a second, much more sophisticated approach, which you put your DNA into the machine and it gives you a list of sequences and the number of times it's found them in that sample, which can then be compared to online databases of what those DNA sequences relate to, and you get a species list from your pond or wherever you've collected your sample. So I'm now going to move. The advantages of using eDNA are that it is very quick. You have a very fast turnaround time with the lab work. It's very low disturbance, so you're not going in with PondNet and smashing up all of the plants and potentially endangering the individuals that are in the water. It is logistically much more simple to undertake a large distribution assessment, so you can go out and collect water from lots of ponds on a single occasion, much more simply than going out multiple times with torches at night or setting traps. It's just easier to organise. It removes some of the need for taxonomic specialists. Obviously, if you're building the DNA libraries, you do need the validation at that end. But in the actual sample analysis, you get your DNA, your sequence read, and you don't need these specialists to approve every individual that's collected. So you're not having to collect rare specimens and preserve them for identification later on. And you can get a sample on for some things you need fewer visits to collect the same amount of data, which reduces the overall cost of sampling. So I'm going to move on to how we're using eDNA. So first, with District Licensing, which is where I work for ARC, it has been instrumental in the setup of the new approach. So firstly, what is District Licensing? So it's a new approach to great crested newts and development mitigation, which is funded through developer contributions. We undertake a risk of impact at a development site. Take that sort of approach. It leads to a reduction in the need for on-site surveys and mitigation. It's not completely removed, but it's reduced. We then replace the on-site mitigation with a landscape scale conservation strategy, which has a strategic approach to habitat creation across the area we're working, rather than putting in a pond on the corner of a housing estate, which is then not managed for 20 years, and anything that's moved into it ends up going extinct. So how are we using eDNA? Well, as I mentioned, it's been instrumental in the setup of these schemes. So this is the South Midlands scheme that we work on. So we've got the Vale of White Horse and South Oxfordshire at the bottom of here. Oxford is about there. Bedford's up here somewhere. And we've got Milton Keynes. This big patch of samples is Milton Keynes over here. To set this up, we randomly surveyed about 650 ponds across the area using environmental DNA. The red dots are the ones which came back positive, black dots are the ones that came back negative. And then we built this data into a species distribution model, which takes both the distribution data and the land cover data, and we could then predict which areas are most suitable for great crested newts in that area, which are coming out red, and which areas are highly unlikely to contain great crested newts, which are the white areas on that map. We also built in known records from record centres in that modelling approach. We then used eDNA to validate some of these areas. So the road we've got running across here is the A420, which runs between Swindon and Oxford. We had this big area of highly likelihood of great crested newt presence to the side of it, but we had no records of great crested newts, either from the record centre or in our analysis through that area. So I went back and I surveyed all of the ponds within this grid square to try and validate the model to see whether it is actually showing us where great crested newts are. Fortunately, I knew the landowner for these ponds here, and she introduced me to her neighbour, and we did find that the model is predicting even where we don't know that great crested newts are present. So we found quite a few of the ponds through this area were occupied, which is really quite encouraging. We are also using eDNA for the monitoring of the schemes. So when we've built new habitats, this is the pond we built about 18 months ago now. This is soon after it was created. We're going back during the breeding season, collecting samples and seeing if it's become occupied by a great crested newt. This pond was occupied within 12 months of creation. Finally, we are using eDNA at the landscape level to assess whether the scheme is having a positive impact on the great crested newt population as a whole. So we've randomly stratified, selected 150 grid squares, which we will be sampling all of the ponds within on a rolling 3-year program. So 50 this year, 50 next year, 50 the year after, then back to the first cohort to look to see whether the proportion of occupancy changes, the number of ponds changes, things like that. Moving on from District Licensing, we also have the PondNet program, which is run by Freshwater Habitats Trust. We now have 5 years worth of data for this. This uses, in 2014, they undertook some statistical analysis that suggested they would need to sample somewhere between 100 and 150 grid squares every year to have a 30% chance of detecting a reasonable change in great crested newt populations. They've set about with funding from DEFRA and Natural England to undertake this work. So every year they've sampled the same 131 grid squares, which is 380 ponds, to look for changes in pond occupancy. Change in pond occupancy, changing in square occupancy, and the number of occupied ponds per square. So I'm rushing a little bit because I can see the clock ticking down. They have shown that between 18 and 35% of grid squares across England are occupied by great crested newts. The first 3 years were fairly stable, and then 2018 there was a bit of a spike, and because this isn't my data, I don't really want to be drawn on why that is. Couldn't really tell you. Similarly, we're seeing about between 7 and 22% of ponds across England as a whole are occupied. Interestingly, the squares without occupancy tend to have lower pond density than the squares which are occupied, but it's very rare to have squares which have complete occupancy, if that makes sense. So it's very rare to have all the ponds within a grid square occupied by great crested newts. So this was, the survey work for this was undertaken by volunteers, coordinated by Freshwater Habitats Trust. So they send the kits out to the volunteers, they collect samples, send them back, and then we have them analysed by one of the labs. This is the first, a world first in using citizen science to collect eDNA samples for a national level distribution assessment for great crested newts. We have found that the volunteers find it very simple to follow the the collection procedures. And what I'm going to move on to now is that it offers a good way of collecting data on uncharismatic species which would otherwise be under-recorded. So what we're moving away from the single species approach I've been talking about with great crested newts to a meta-barcoding approach, where you collect your sample of water, send it off to the lab, and then they get the DNA sequences out of the sample, which is then run across online databases, so you end up with a species list and the number of times that has been picked up within the sample. We've been doing this in the New Forest, specifically targeting fish, and this is with the help of Nature Metrics, who are sponsoring the conference. We have been sampling streams which have been being surveyed by the Environment Agency for about the last 30 years using traditional fisheries-based methods. And we have found that all of the species the Environment Agency have been finding have been coming up in our analysis, with the exception of 2, and these haven't been found since 1990, so we're fairly sure that they're not there anymore. We did find 2 additional species that have never been found by the Environment Agency, the spined loach and the bullhead, which is quite interesting, and it suggests that these eDNA sample surveys are at least as sensitive as conventional fishing methods, even when you're using volunteers to collect them. Volunteers seem to enjoy collecting these samples, even though they're not directly seeing the species that they're looking for, and they do this well. And that combining citizen science with environmental DNA metabarcoding is a very effective way of collecting large-scale data sets, as we were hearing about earlier. Interestingly, in those samples, we have found things like kingfisher using the stream, so it's not just fish which are permanently in the water, it's things which are only temporarily visiting. Worryingly, for a lot of the ponds in the New Forest that were sampled, there's a very, very worryingly low number of instances of water shrew were picked up. And Naomi had told me another one to say, but I've forgotten what that one is. So these are a few of the projects that we have coming up that we are hoping to be using environmental DNA for. So we have quite a big project trying to restore fens around Oxford. When I say we, Freshwater Habitats Trust do. So they're going to be trying to use eDNA to look at the diversity of Diptera, so fly species. We also have another one with the National Trust that is going to be looking at the amphibian and fish diversity in their ornamental lakes, which we are hoping is going to be very, very useful link with their properties there. And so that's all I have, and I see the screen's just popped over to 1 minute. So.